Hello and welcome to this Infection Basics video. I'm Dr. Alid Roberts, and in this video we're going to look at some specific exotoxins known as the AB toxins and then work through this with a key example in the form of the cholera toxin. And one of the first things we need to ask ourselves are, what are the AB toxins? Well, in bacteria, AB toxins are a special type of exotoxin, meaning they are proteinaceous and secreted from the producing bacterial cell. Now, AB toxins are a unique group of toxins that have a characteristic two-component system that disrupts important cellular processes, and each component of the system, so the A subunit and the B subunit, have distinct functions. The A subunit is the active subunit and is enzymatically active, meaning this is the part of the toxin that disrupts cellular processes within the host cell. The B subunit, on the other hand, is the binding subunit with no enzymatic activity. The sole purpose of this subunit is to ensure the effective delivery of the A subunit into target host cells. And so an important aspect of AB toxins is that both are required. If a cell is to only produce the A subunit, then the toxic effector protein would have no mechanism to get into the target cell, essentially rendering it useless. Then on the flip side, if a cell was to only produce the B subunit, then the binding protein would associate with the host cell receptors, but they wouldn't have an effector protein to deliver into the host cell, again rendering it useless. Now, all bacterial AB toxins follow this two-component structure, so let's have a look at one of the most well-used examples, and that would be the cholera toxin, produced by Vibrio cholerae and used to induce an electrolyte imbalance within the gastrointestinal tract, leading to diarrhoea. Now, a key question is why? Why exert energy and resources as a bacterial cell in order to produce the cholera toxin and induce diarrhoea within a host? Well, put very simply, it is to increase the chance of transmission from one host to another, as Vibrio cholerae is commonly transmitted via what we call the fecal-oral route and the consumption of contaminated water. Now, the disease resulting from this toxin is an extremely pale diarrhoea known as rice water stool, and this is due to the rate at which fluids are leaving the body and has become a hallmark feature of the disease. So let's have a look what is going on here. And so the A subunit of the toxin is 28 kilodaltons in size and has two functional domains named A1 and A2, and this is obviously the toxic effector protein. Then on the flip side, we have the B subunit, which is composed of 5 11 kilodalton subunits, each of which are identical with a single domain and associate with one another to form this pentameric ring structure, which is crucial to its function as a receptor-binding protein. And so the B subunit is responsible for binding to intestinal enterocytes via the ganglioside GM1 receptor. This allows delivery of the A subunit into the cytoplasm of the host cell, where it undergoes a little bit of processing, becoming activated and allowing it to bind to G proteins. Now, this binding alters biochemical processes within the cell, and in this instance it causes the overproduction of cyclic AMP from ATP, so converting all readily available ATP into cAMP. So let's have a look at this entire process in a little more detail. So here we have our host enterocyte cell lining the gastrointestinal tract, and within these is a GTPase enzyme known as the G protein. Now, one of the main functions of this protein is to stimulate the activity of adenylate cyclase, but it only does this in response to hormone signals. And so when it receives a signal, it activates the G protein, resulting in a sort of conformational change that allows it to associate with adenylate cyclase. And this binding activates the adenylate cyclase enzyme, which converts ATP into cyclic AMP. And due to the nature of the interaction between the two, after a short period of enzymatic activity, they dissociate from one another, resulting in low intracellular concentrations of cyclic AMP being produced. Now, to complete the process, cyclic AMP interacts with protein kinase A, and like all kinase enzymes, it's responsible for phosphorylation. In this case, it leads to the phosphorylation of membrane transport proteins such as the cystic fibrosis transmembrane conductance regulator protein, or CFTR for short. Now, in this phosphorylated state, the CFTR protein acts as a channel, allowing chloride ions to move from the cytoplasm of the enterocyte into the gastrointestinal lumen. And because we have low intracellular concentrations of cyclic AMP, we have low phosphorylation rates of the CFTR protein, resulting in the controlled release of chloride ions from the cell. So that's what's going on under standard or normal conditions without infection or disease. But what about in the presence of Vibrio cholerae that is producing the cholera toxin? Well, during infection, we get quorum sensing and the production of the AB holotoxin, which we can see here. Now, when in sufficient quantities, it binds to ganglioside GM1 receptors on the surface of our enterocytes. And at this point, the holotoxin receptor complex is internalized via endocytosis, and to cut a long story short, it is trafficked to the Golgi apparatus, followed by the endoplasmic reticulum, so a sort of reverse trafficking within our cells. And it's in the endoplasmic reticulum where the A subunit is released and processed, cleaving it into two to form the A1 and A2 subunits. And it is the A1 portion which is catalytically active, and this unfolds and is translocated into the cytoplasm of the enterocyte. And in this state, it is able to interact with our G protein, which activates it, resulting in a sort of conformational change that allows it to associate with adenylate cyclase. This in turn activates the enzyme which converts ATP into cyclic AMP. However, unlike last time, the presence of the cholera toxin stops the dissociation of the G protein from adenylate cyclase, meaning we get the continual uncontrolled conversion of ATP into cyclic AMP. Now, this results in relatively high intracellular concentrations of cyclic AMP. Now, to complete the process, cyclic AMP interacts with protein kinase A, leading to the phosphorylation of the CFTR membrane protein and the movement of chloride ions from the cytoplasm into the gastrointestinal lumen. Now, because we have high intracellular concentrations of cyclic AMP, we have high phosphorylation rates of the CFTR protein, resulting in the uncontrolled release of chloride ions from the cell. Now, when this happens, chloride ions build up in the lumen, and to counter this, we normally get sodium ions following, which then leads to the movement of water into the gastrointestinal lumen, and it is this which forms the pale rice water stool we talked about earlier. And so to summarise, cholera begins when Vibrio cholerae is ingested via contaminated water and survives passage through the stomach to colonise the small intestine. Then they secrete the AB cholera toxin into the intestinal lumen, where it binds to GM1 ganglioside receptors on our intestinal enterocytes. This allows the A subunit to enter and permanently activate adenylate cyclase, causing a sharp rise in intracellular cyclic AMP levels, which interferes with the cell's normal biochemical processes. And the result of this interference is elevated cyclic AMP levels, which causes an electrolyte imbalance by keeping CFTR chloride channels open, driving chloride secretion into the gut, followed by sodium and large volumes of water. Then this manifests itself as rice water stool, as profuse watery diarrhoea leaves the body, leading to rapid dehydration and electrolyte loss. And with that, we come to the end of this video. I hope you found the content useful, informative, and most importantly, easy to understand. Thanks for watching, and I hope you have a great day.